Good morning, MacLaren. Today I want to talk to you about viruses. Viruses are so fascinating to me. They are these particles that we can't see, and yet they impact every living organism. A virus is a submicroscopic infective agent, which just means that it's too small to be seen under a light microscope. And it's an obligate intracellular parasite, which means that it has to be inside another cell, which we call a host cell, and it uses that host cell's energy to carry on its processes. Different viruses have different structures that allow them to attach to and then get inside of different host cells. This is why there are some viruses that can infect humans but not dogs, or some viruses that can infect a rabbit but not a bird. Some viruses that you may be familiar with, we're most familiar with these harmful viruses. They're harmful to their hosts. So parvovirus is a virus that infects dogs. Influenza, hepatitis, rabies, herpes simplex virus, which causes fever blisters, dengue fever, which is caused by the dengue virus. But some viruses are very beneficial. So mammals have a host of viruses all throughout their intestines that protect them against harmful bacteria. And they do this because there are some viruses called bacteriophages that only infect bacteria. So if you ingest a bacteria that might be harmful for you, then this bacteriophage is going to take care of that bacterium before it is able to harm you. So let's talk about the structure of a virus. A virus is pretty simple. So in the very middle, it has genetic material. Sometimes it's single-stranded, sometimes it's double-stranded. That just depends on if it's DNA or RNA. Surrounding the genetic material, there's usually either a capsid, which is made of proteins, or an envelope, which is made of a lipid bilayer, which is has the same composition as cell membranes. And then in that envelope or capsid, you have different proteins that your body would call that we call antigens when we're talking about an immune system. And that is what an immune system would recognize as foreign. And then that is what antibodies would attach to if it has antibodies, or what immune cells would recognize to fight off the bacteria. So what do viruses do? Well, mainly they just infect a cell, replicate, and infect more cells. So in order to do this, they need to get inside of a cell. So it's going to attach to the outside of a cell, and then it's going to enter the cell, and it has to have just the right components on the outside of the virus to be able to attach to and get inside of a specific cell. Once it's inside the cell, the either envelope or the protein capsid are going to come off of the genetic material. The genetic material is then going to be replicated using the host cell's replicative enzymes. So the host cell has the tools, and the host cell is going to make a lot more genetic material. And then the viral RNA is going to be recognized by the host cell's ribosomes. And then the RNA is going to be read and then turned into proteins. Then you have more viral genetic material and more viral proteins, and they come together. It's called assembly. And you make more virions. That's a unit of a virus. And then those virions explode out of a cell. Sometimes it's graceful, sometimes the cell actually explodes. If there was a lipid envelope, a lipid bilayer envelope, then those virions, as they exit the cell, are going to take some of the lipid bilayer from the host cell's membrane, and that's going to compromise the integrity of the host cell, and then that cell is most likely going to die. So you start with one virus, it infects a cell, and then it makes a lot more viruses. And so this spreads exponentially. So this becomes a race. Which is going to happen first? Is the virus going to replicate and make more viruses that then do a lot of harm to the host? Or is the host going to recognize that it's a virus, create antibodies, have an immune response, and eliminate all of the viruses first? So that's the battle on what's going to happen. So if you're infected with a virus, then your immune system does its whole process. The end result is your body saving basically a program, we call them antibodies, on If we see this again, we have a plan and we'll be faster and more efficient and we will take over the virus before it can make us sick. But then why can you get sick with the flu several times in your life, and maybe several years in a row, or maybe even twice in the same year? If you're making antibodies, how can you get sick twice? Well, the reason for that is mutations. So as a virus replicates, it's copying genetic material. And the viruses that we get sick with the most often and several times in our life usually have RNA genetic material. And so when RNA is being copied, there's not a proofreading mechanism. So there's no way to go back and make sure, for the virus to go back and make sure that everything is correct. So if there's a mistake in the replicative process, then that mistake stays. And if enough mistakes accumulate over time, then there might be a mutation that affects the structure or the shape of a protein. And remember, those proteins are what stick out of the virus, and that's what our immune system recognizes as foreign, and that's what our antibodies bind to. So if those antigens change, then our antibodies don't work for that new virus. So here is a slide of just a really quick how a change in genetic material can change the shape of a protein. So up here at the top, we have the DNA, and then this shows the different protein sequence, so the amino acid sequence. And if you see from the top to the bottom, there's a switch. In one place, there's a T, and then in the other place, there's an A. So that's going to be the mutation. And the reason that matters, this is not a lesson in mutations, but the reason that matters is look at those pictures of those cells. On the top, you have normal blood cells. And on the bottom, you have abnormal blood cells. They're called sickle cells. They stick together. They don't carry oxygen the way that they're supposed to. And they just generally affect whoever has sickle cell anemia. They have different symptoms, and it's all because of this one mutation. So let's go back to viruses. So how does this happen in viruses? So in a virus, mutations happen slowly over time. And after enough time, there's going to be enough mutations that cause a difference in the proteins. So these proteins are what's sticking out of the cell. And if you notice from the top to the bottom, you have purple triangles on the top, and on the bottom, that surface protein is now a purple oval. But you still have other things that are kind of similar. So your body kind of recognizes it, but not really. And so you might get sort of sick, but not super sick. Okay? That's called antigenic drift. So those are antigens, they're slowly changing over time. They're drifting. And this happens slow enough that in a community of people, immune systems can adapt quickly enough to the slow changes that it looks like a different flu shot every year, a slightly different strain. If you get the flu, well, you've had the flu before, you might not be hospitalized, you might just feel pretty crummy for a couple of days. That's antigenic drift. Let's talk about the Spanish flu. So with the Spanish flu, the flu had been around, people had gotten the flu before, but with the Spanish flu, there were two different strains of flu, so two different flus. And they infected the same cell, and this can happen in a human or a different animal. Sometimes birds, sometimes pigs, that's why you hear bird flu, swine flu. You have two different types of flu, and they infect the same host cell. And then that envelope and that capsid, they go away, and then you just have strands of RNA. And it's this mix and match situation that happens. And then you have this reassembly of a new virion that doesn't look like either one of the other viruses. So now you have a completely new strain of flu. And if you notice in this picture with the antigenic shift, the antigens, those surface proteins, are totally different. And so when antigenic shift happens, then you have an entire population that their immune systems don't recognize this. So even if you've had the flu before, this is so incredibly different that it's unrecognizable to your immune system. And so then you have lots of people getting sick really quickly. So I just hope that this shows you a little bit about what viruses are, how they work, and then why some mutations are more impactful for others. Thank you.